Chapter 9, Mutations and Genetic Engineering. So by the end of this chapter, there's 3 things that you should know. You should know what mutations are, what their impact is, and how they occur. How can genetic engineering be used to treat genetic diseases, as well as if all mutations are harmful. So let's start off with what a mutation actually is. So a mutation is a change in the nucleotide sequence of a DNA molecule. This chapter focuses pretty heavily on sickle cell anemia because that is a genetic mutation that we're looking at specifically in terms of how mutations occur and what can be done about them. But mutations, like I said, a change in the nucleotide sequence of a DNA molecule. So you can see here kind of the process that it goes through. This is actually for sickle cell anemia. So you can see what happens when it's normal and what happens once we have the mutation. Now you can see here that mutations aren't big, but they have a huge effect. And we'll go into different types of mutations, but basically you can just be changing possibly one nucleotide. So you can see here we have 1 point mutation that's occurring. So we have CTC in our normal expression and then CAC here. So we've just switched the A and T in this one, and it results in sickle cell anemia, which we'll talk a little bit more about. But just know that mutations can be something really small that really lead to a big effect. So importantly, how do we see mutations come up? What happens with them? So like I said, it's that change in the sequence of the DNA. So there's a few different ways that it can happen. And one is obviously through genetics, but also you have other things going on as well. And with these mutations, when you're seeing them, it's basically just a different version of the same gene. And when that is produced through mutations, we call them alleles. So you'll get into that a little bit more in the next few chapters as you're understanding what genes are, what alleles are, and how you can see the differences in them. But basically, when we're looking at a gene, we're looking at something that is responsible for some sort of protein expression. And different alleles means that there's different versions of it, essentially. So these mutated ones are a different version of it, or a different allele. Okay. And if we get too many mutations, or if it's affecting some sort of normal functioning of the cell cycle, that's when we can get into cancer as well. Okay. Cancer we'll talk about in the next chapter, so just kind of keep that in the back of your mind for right now. So again, mutations occur after you've altered the sequence somehow. So it can change the coding region of a gene, which might mean that the protein that we make after that will not necessarily be functional. So you can see here we have a normal BRCA1 protein synthesis, and then we have one, it's called an insertion frameshift mutation happening here. And what it's doing is it's changing the transcription into mRNA into something different. Okay, so instead of making these amino acids, right? So say we go to UCG for our mRNA, which would make serine. Instead, now we're making tyranine. And then it's affecting the ones subsequently after that as well. So instead of making a normal BRCA1 protein, now what we've done is we've made a non-functional BRCA1 protein, which means that we're not going to be able to do the things that we needed to do with that protein beforehand. Sometimes it means it's not functioning. Sometimes it means that it's making something completely different. So there's lots of different types of mutations, and they have different effects. So again, original DNA sequence, you would see no mutation, right? So here's some examples that you can read through. And I think that this is actually the chapter 10 class activity because chapter 9 and 10 are switched. But DNA has all you can ask for, right? So that's what our original DNA script says. Now we come down here, if we have something like a point mutation, which is where we just have one thing changed, one nucleotide changed in our sequence. And you can see that it's highlighted here in red, right? So one is we have a silent mutation. A silent mutation means that we do change one nucleotide to another, but it doesn't actually affect the amino acid sequence. This is arguably the best kind of mutation you can have. It means that although you've changed something, you're still going to have normal function. You're not really going to see any negative results coming from this. Okay. On the other hand, you could have a point mutation that is a missense mutation. So this is changing one nucleotide to another, just like you did up here, but this time, now we've changed it to a completely different amino acid sequence. And that means that we're going to change the protein shape and function. Okay. We could also have a point mutation that's a nonsense mutation. Again, only changing one nucleotide, but this time it's producing an early stop codon. So remember, our stop codons are telling it to stop, basically stop reading and stop producing the RNA and DNA replication. But stop producing the RNA replication, which would also change the protein. It's going to make it too short and therefore not functional. So 3 very different kinds of mutations that you can see with just changing one nucleotide here, which is a point mutation. We can also have frame shift mutations. So this is where we're shifting our nucleotides one way or another. So we could do something called an insertion mutation, and that's where we're inserting one or more nucleotides. So it's going to shift the reading of the frame of every codon after it. So you can see here DNA has all U, right? So instead of changing this one, now we've just added an extra Y. So what that did was it changed this amino acid, and then it's also going to change this one, and this one, and this one, and so on and so forth. So we're going to have several modified sequences that could be severe and, again, make the protein not functional. We could also have a deletion mutation. So instead of inserting, now we're just going to delete one. So DNA has all, now we've deleted that second L. So the Y is moving over one, and it's shifting the reading of every frame for every codon after this as well. And again, we're severely modifying several different sequences that will, again, make the protein not functional. Last but not least, we can also have rearranged DNA mutations, okay? So this comes as either an inversion or a translocation. An inversion just means that a group of DNA nucleotides are flipped, right? So DNA has all U can. Our C and our U got flipped here, which is making 2 different amino acids here. So that means it's going to change the protein shape and function. We might be making an entirely new protein. On the other hand, if we have a translocation mutation, this can move the segments of DNA from one chromosome to the other. Okay, and what this does is it fuses portions of different genes together. So this is like a pretty significant mutation. Not that the others necessarily weren't, but translocation mutation is shifting quite a bit more. And because of that, you're going to see a significant change in protein shape and function, possibly non-functional, or you're just going to get an entirely different protein altogether and not make the one that you had originally intended. Okay, so again, these mutations can arise. They can occur during DNA replication. They can also happen because of different environmental insults. The other way that they can happen, obviously, is inheritance. So inheritance, you could inherit this from mom or dad, and that's the case that we see with sickle cell anemia, right? We have a mutation in the beta globin gene, and that can be inherited from either parent. And if it's inherited from both of them, then you're going to have sickle cell disease, okay? So we'll get into this more in, I believe, chapter 11 and 12, where we're talking about if you're a carrier or not, and some of you have already been exposed to that a little bit. But in this case, we can see both parents are carriers. They're both carrying sickle cell, but because it's a recessive disease, they do not have it. But they're both going to pass down their recessive alleles to their offspring, right? And not to everyone. There's only a 1 in 4 chance that one of their kids will have it. But let's say they have 4 kids and one of them does actually get that, then that kid would have sickle cell disease. So that is one way that mutations can arise and can continue to live in different populations. DNA replication errors are also a way that mutations can arise, and it's actually way less common than inheritance or mutagens, which we'll talk about here in a second. But mistakes do happen during DNA replication. Your DNA is constantly replicating, and it's making lots of new DNA and going through transcription and translation. And because of that, things can happen, right? Most of the time, we correct these mistakes by repair enzymes and cell cycle checkpoints. But we do still get a mutation on average that's happening every 10 billion base pairs, and that one will get replicated. It could be something like a point mutation, that doesn't do anything. It doesn't change your protein shape or function, but it could, on the other hand, be something that's pretty significant. And last but not least, we have mutagens. So there's lots of different components of the environment, our food, and even our cells that can cause mutations, and things that we can do to our body that aren't optimal, right? So like receiving radiation, if you're constantly getting x-rays, UV radiation is obviously terrible. Chemicals, pollutions, pesticides, smoking, alcohol, char. And then there's also infectious agents like some viruses like hepatitis C. We're also seeing that to some extent with COVID, although we don't know the long-term effects and what mutations that we're seeing long-term with COVID. There is a possibility that we're going to see something similar like we do with hepatitis C. And then also different cellular reactions that can happen as well. So as far as DNA replication errors, you go through this a little bit more in the next chapter, but just know that there's different cell division cycles that happen for your DNA to, not just to replicate, it's replicating anyways, but to actually create new cells and synthesize new cells. We either go through mitosis or meiosis. Mitosis for your body cells, meiosis for your reproductive cells. And as it's going through these, there are little checkpoints that say, hey, did everything go okay here? Yes? Okay, then we're going to keep going. No? All right, then we're going to go backwards and we're going to fix it. Okay, so if something happens to those checkpoints, or if it misses it, that's when the DNA replication errors are going to happen and we're going to have a mutation, which again may or may not be significant in terms of protein function. As we're going through this, if something called apoptosis doesn't happen, which is programmed cell death, then you're going to accumulate enough of that DNA damage to interfere with those checkpoints. And that usually results in cancer, sometimes in different mutations and or both, which again, we'll get into more in the next chapter, but just kind of keep that in the back of your mind. Okay. And again, a very small proportion of these are due to mutations that are inherited. So we see this with the breast cancer genes that we'll talk about again more so in the next chapter. But just because you inherit a breast cancer gene doesn't necessarily mean that you're definitely going to get breast cancer. It just means that you're at an increased risk of inheriting breast cancer. And any family member can carry different mutated genes, but you do see them happening more in certain ethnic groups. And part of that is because we tend to, depending on where you are in the world, you tend to be dating, marrying, producing offspring with other people that are geographically somewhere that you can access, right? Which in a lot of places in the world are people that are near you. And we're not talking about like inbred family or anything like that, but people in your population in general where some of these things might be at a higher prevalence, you would have a greater chance of passing that on to your offspring as well. So something like sickle cell, this actually does correlate really well with different regions that have high malaria. And the reason isn't necessarily just because it's a geographically isolated thing. It's not because we're just reproducing with people in those areas, but it's also because it confers a genetic and evolutionary advantage. Why would sickle cell be something that could ever be advantageous? It sounds terrible, right? We're at higher risk for blood clots. We're not getting quite enough oxygen. We've got lots of different issues. We're increased risk for stroke, but it does actually help with malaria. Right? So ideally we want to not that you necessarily want to have sickle cell, but if you have sickle cell anemia, it does help with the frequency of malaria. And you can see that the frequency of sickle cell mutation, it's selected for. So it's something that makes you more likely to survive and thrive in these environments, which is why we see really high prevalence of the sickle cell mutation in areas that also have lots of malaria. Okay. So again, the last one on our list, the mutagens. So a mutagen is just a chemical or physical agent that can actually damage DNA. And it does that by changing the nucleotide sequence, just like any other mutation. And again, we'll learn about this more in the next chapter, but a mutagen can also be a carcinogen. Carcinogens are substances that cause cancer. Most carcinogens, not necessarily all, but for your purposes, I would say all carcinogens are also mutagens, but it doesn't go the other way around. So you can have a mutagen that is physically damaging your DNA, but is not necessarily likely and or going to cause cancer. On the other hand, if you have a carcinogen, like what you see on almost all labels of everything that this is known to cause cancer in like the state of California or whatever, that's a carcinogen, is also a mutagen. If it's a carcinogen, it is definitely also damaging your DNA or has a high likelihood of being able to do so. So going into sickle cell disease a little bit more, again, this is a point mutation. So this is a single change in the nucleotide sequence of one gene. So again, we're looking here, we've switched the A and T here in the DNA. When we go to transcribe it into our RNA, that's obviously changed as well, which is then going to change our amino acids, and then is going to change our protein. So what it does is it alters the shape of this hemoglobin. So instead of having this nice little circle, it's going to alter the shape of hemoglobin where it's more sickle cell shape. Okay, so these abnormal hemoglobin actually block blood vessels and are not able to carry as much oxygen as our normal hemoglobin. Okay, so this is the protein beta globin. That's what is encoded for in this specific gene. So if we have normal beta globin expression, we are quote unquote normal, right? If we have that point mutation occurring, that's who is diagnosed with sickle cell. And again, sickle cell, unless you're living in an area with high prevalence of malaria, is not really a great thing. It can cause things like anemia, extreme pain, blood clots, stroke, and sometimes even death. So it's not something that anyone really wants to live with. It does still confer an evolutionary advantage in some areas, like Africa, and that's why it's still around. But just because it doesn't have an evolutionary advantage doesn't mean that it's going to be necessarily super easy to kick out of all other populations either. So one of the things that your book talks about is gene therapy, and gene therapy is not super new. It has been happening for quite a while, although it is getting a lot better, and it's getting a lot better really, really quickly. This is something that's really been happening, I think, since the 90s, but you hear horror stories or, that it may or may not work, and there's different things that have been tried, and now it's really becoming a little bit more prevalent, and we're starting to see more positive results from it. And gene therapy is just correcting or replacing genes as a treatment for some of these genetic diseases. And the one that we're focusing on here, obviously, is sickle cell anemia, or sickle cell disease. Okay, and so what they do basically is we take someone's blood, the person with sickle cell disease, and these people do have a mix of normal and sickle cells. They're not all sickle cells necessarily. It just means that a lot of their expression is changing to sickle cells. So we take their hematopoietic stem cells. A stem cell is just an undifferentiated cell, so it can become anything we want it to, basically, which is why stem cells are really valuable. You've probably heard of stem cell research, and you might have heard of it being controversial or anything like that. Honestly, from a purely science point of view, it really shouldn't be that controversial. Stem cells are great, and there's a lot of things that we can do with them if we are working with them in the right way. But hematopoietic, that just means that it came from the blood. Okay, so we're going to take these with a mutated beta globin gene, from that person that has sickle cell disease. And then there's two different kinds of gene therapy that are discussed in your book. This is the first one. And what they're doing is they're actually injecting a virus that has altered to carry the normal beta globin gene. So when the stem cells are infected with this virus, receive that normal beta globin gene, it's getting stitched into the stem cell chromosome. We're going to grow them in large numbers, going to grow a ton of them, and then we're basically going to put them back into the patient where they're going to be able to produce red blood cells, normal red blood cells. So that is one version of gene therapy that we're seeing. Another version is a little bit different, and it's kind of going for a different approach. So this one is basically going for a genetic switch. So there's different types of hemoglobin that we produce in our lifetime. So before birth, we are making something called fetal hemoglobin. And then we start making adult adult hemoglobin in general at a later point. And they are two different, they're different in almost every way, although they are doing basically the same function. And this fetal hemoglobin is not affected by the sickle cells. There's no disease symptoms or anything. And you can see this is before birth, right? And then we hit birth, and that's when, if you have sickle cell disease, that your fetal hemoglobin is going to decline, and your mutated adult sickle hemoglobin is obviously going to increase because that's primarily what you're making. You are still making a little bit of fetal hemoglobin, but it's not quite, it's not very much. And so after you have a period of time, usually only a few months, where you're making primarily sickle cell or sickle hemoglobin, then you're going to get the onset of symptoms, right? And for most people, I don't think that they're really doing gene therapy on any younger adults or kids at this point in time, although technically they could, at least in the future. But you can start gene therapy, and what this gene therapy is doing is, again, we're basically switching on the gene that will instead make fetal hemoglobin, so that hopefully we can make more fetal hemoglobin and it can kind of take over for that sickle hemoglobin, so we're not getting any of those symptoms. So after this gene is turned on, that's the hemoglobin that they're going to make, and we're all well and good. And some argue that this is a better approach, some argue the other one is a better approach. Ultimately, they're both kind of going after the same thing. The people arguing that this is a better approach are arguing that because you're introducing your body to something it's already doing, right? We're not having to inject it with a different virus that's doing something different. We're just basically turning something on that almost was like long forgotten. And once we turn that on, then we can kind of take over and have your body naturally do its thing, versus, again, injecting something somewhat foreign into your body. But they're both seemingly producing pretty good results so far, so which one is better, only time will tell. And, again, they're both producing positive results, where people are seeing, an offset of symptoms, which is great. There's one other thing that could work, and it's called gene editing. So this is a way to change the sequence of a gene, and the way that we do it is with something called CRISPR. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is why we just call it CRISPR, because who wants to say that over and over? And this is something that's fairly new. It's become a lot more popular, I'd say, in the last decade or so, especially in the last few years. But it's basically a genome editing tool, and it's based on something that happens in bacteria. And it's essentially like these molecular scissors, they're bacteria scissors, that can go in and cut out the things that we don't really want. The advantage of gene editing over some of the other gene therapies we just discussed is that it's really quick, it's easy, and it's cheap. And those are obviously all really great things if you're trying to introduce them to more of the population. So the way it works is that there's basically two main components for CRISPR. So first, what we do is we basically introduce an enzyme that is going to cut the DNA and a small piece of RNA that serves as the guide for the enzyme, directing it to a specific sequence of the DNA. Okay, so this is just so that we can modify a target sequence. So once we add that enzyme and it goes in, it's going to specifically bind to that target sequence and cut the DNA exactly where we want it to. Once the DNA is cut, then what we can do is we can modify or add a specific DNA sequence to that location. So we're going to cut it here, right? And now we can add something that would make normal protein, like non-sickling beta globin. So basically what we're doing with CRISPR is, at least in principle, is we are correcting the genetic mistake that causes the disease in the first place. So you're kind of going back to the drawing board, right? You're going back to the very beginning of it and you're looking at where the mistake was made and you're correcting it there instead of later having to do a gene therapy. Now we've just done gene editing. So once we've edited the gene and we've mutated the, this version in the person's blood stem cells, what we can do is that person will now only be producing healthy hemoglobin, so they wouldn't experience any of the other symptoms that they would have had previously. So it's pretty cool technology. Honestly, it's still a little bit of ways. They're not quite to the point where they're doing this with sickle cell anemia patients. They are doing it with other things and they're testing it in lots of different animal models, but it is still relatively new and not 100%, I guess, available on the market, at least definitely not in the US. So is gene editing or gene therapy a permanent fix? Not really, right? It is hopefully a somewhat permanent fix for that individual, but we are still only changing a subset of our somatic cells. Somatic cells are our body cells. They're our non-reproductive cells. They're the ones that are going through mitosis, right? But this mutation is still going to be occurring in the germ cells or your reproductive cells. These are the ones going through meiosis. So that means that although you may not be experiencing sickle cell anemia if you once had this disease, because we've, gone through either gene therapy or gene editing, you can still pass this to your children. Okay. Gene editing in embryos is potentially possible. It is very controversial, mostly because, anything with embryos is really pretty controversial. I think that's kind of what I alluded to earlier with stem cells as well, is you don't have permission from that embryo, who will then become, a human child and then later on into an adult. You don't have permission to edit their genes, basically. And so this is kind of controversial, not just for things like sickle cell anemia or other diseases that are really either life-threatening or really affect quality of life for these, fetal embryos, but could also be other things, getting into editing their genes so that you can essentially pick out different qualities or things that you want your child to showcase, basically, different phenotypes that you might be able to choose and that kind of thing. So pretty controversial. There are some people who've done it. I think your book talks about a Chinese scientist who did it a few years ago, and everyone was pretty salty with him. But it does have some potentially really neat advantages that it could confer, but again, ethically, it can get a little bit blurry. Speaking of ethically and a little bit blurry, I just wanted to touch on regenerative medicine. This isn't in your book, but it is something that you might have heard about before, or you might hear about more in the future. It is also kind of becoming more and more popular along with things like gene therapy and gene editing and CRISPR. And regenerative medicine is basically, it's not just growing human organs, but it's using things that our body uses to constantly regenerate to do things, essentially. And we're doing this by using stem cells. And again, stem cells are just immature cells that can divide and differentiate into specialized cell types. So we can grab, like what we grab those hematopoietic stem cells for our sickle cell gene therapy. That just means it came from the blood. We can grab stem cells that come from some other part of the body, or possibly from placenta or something like that. And we can make that grow into, we can have that differentiate into, say, a kidney cell, if that's what we're needing. So this is how scientists are using it to, grow other organs or something like that. And what they'll do is they'll isolate cells from a patient's tissues, and then they basically coax them to divide and populate a scaffold that resembles the organ that we're looking for. So this is something that's really difficult to do with something that has lots of different cell types, like a kidney. A kidney has lots of different layers, lots of different cell types. But if we're talking about doing something like a bladder is actually really easy to grow outside the body, because it's essentially like this balloon that only has a couple main tissue layers and only consists of a few different specialized cell types. So bladders are pretty easy for us to make. Some other organs are obviously pretty difficult, but what they'll do is they'll take that diseased organ, remove some tissue from it, have the cells divide and culture, and then they'll put them on some sort of scaffold that can be grown outside of the person's body. Sometimes they do it on the person's body, and then they basically go back into the patient and restore the bladder or whatever organ it was that they used regenerative medicine for. And again, if we're doing this from the patient, then we see no immune rejection, which is awesome, and an improved urinary function in terms of the bladder. But there's other reasons that you could use this as well. And again, it's just taking stem cells and making them into a more specialized type of cell. There's a few different ways that they could do this. Again, they can do the biodegradable scaffolds, and they can grow things like even an ear here. There's a crazy picture that I want to say is popular, but maybe it's only popular with people like me, of them growing an ear on like I think the back of a rat. So there are many different places that you can grow new tissues or organs that you want to transplant into patients. And ideally, we want to make sure that we're getting stem cells from the patient so that we don't have any sort of rejection of the immune system. But we also can use things like therapeutic drugs that stimulate stem cells in the body to repair certain tissues, right? There are certain tissues that are constantly regenerating, but some tissues and cells regenerate far less. So like our brain cells, right? Our parents always warned us, wear your helmets, protect your head, don't get concussed, that kind of thing. It's because our brain cells are not regenerating at a very fast pace, if at all. Once we lose brain cells, we lose them. Whereas if we lose skin cells, those regenerate really quickly, right? We can heal really quickly if we are quote unquote normal. But brain cells, some other cells throughout the body and in different organs and tissue systems do not regenerate that quickly, if they regenerate even at all. Okay. So again, there is one other way that you could do this, is that you could use cadaver organs. And basically what they do is they kind of bleach them, essentially, in finer terms, and take away all of the cells from that cadaver organ donor, and then they will inject it with cells from the stem cells from the patient, and then they'll transplant it back into the patient that needed that organ. So there's a few different things that you can do with regenerative medicine. It is kind of a really cool up and coming area, but still kind of like gene editing and things, a little bit controversial. And I think it's, there's a lot of things ethically that go into it. And, even like cadaver organs, we have organ donors and things like that. But, there's a lot of components to it ethically, and morally that have a lot of objection and pushback from people. But there are a lot of really great things that we could do with it. So it'll be interesting to see where gene therapy, and regenerative medicine go in the future. There's, like I said, a lot of really cool things that can happen and a lot of challenges that come with it as well. But that's all for chapter 9.